In this video, we'll explore how to use the pathologic inference component of Pathway tools to create a new Pathway Genome Database. The simplest way to create a new Pathway Genome Database is by running Pathologic at the command line using a Unix command such as this one. I'm running here on a Mac using the terminal window to start the program. And what I'm going to do is to run Pathway tools across a single GBFF file for Acinetobacter baumannii that I've downloaded from NCBI. We can launch the software as follows, and it begins by creating a new empty PGDB and then loading in the GBFF file we just specified and running multiple different components of Pathologic on it, such as the pathway inference, the transport inference parser, creation of a cellular overview diagram, etc. And this whole process is fully automated, takes about 6 minutes, and gives you a draft PGDB. You can run the command line Pathologic on both single files and on a directory of files. You can run it on multiple directories of files to create databases from multiple genomes in one run. You can run hundreds or thousands of genomes through the software. But we're going to focus mostly in this video on running Pathologic interactively through a graphical user interface. And one thing to note about both the command line based and the graphical user interface based execution of Pathologic is that as it runs, it creates various report files. And here's one listed right here. Creates various report files that log what the software has done. What inferences has it made and why? Has it found error cases or ambiguities that it was unable to process? It's worth looking at these different report files that are stored in the reports directory of the new database that was created. So every new database that's created gets a new directory. Now, to launch the interactive version of Pathologic, we start at the Pathway Tools Navigator, and then we go under the Tools menu and select Pathologic, and we get the resulting window. And to create a new database, we go under the Database menu and say Create New. And the resulting dialog has several different portions. It's only the upper portion that's required, and in fact, the software actually fills in most of these fields from the input file itself. So let's select a file. We'll click here to select our input GFF file or GBFF file or GenBank file. I can simply type the file name here, or I can go through the interactive mouse-based selection. Okay, so it's recognized the file. It's extracted the project identifier using the refseq identifier from the file and put that as the default value of the organism project identifier. And we've created a version of the database, and we're going to store the database as a file, the new database that's created. It can also be stored in a MySQL database if you've configured a MySQL server appropriately. Now in this next required section of the file, it asks for taxonomy information about the organism that we're processing. And again, the software has read the file that we just provided and extracted the genus, species, and strain. So we don't actually need to do anything additional here. We can enter a genome source. This would be, for example, it could be the name of the organization that sequenced the genome. Often we'll fill this field in when we have multiple genomes available to Pathway tools for the same strain of a bacteria, and we need further differentiation between those same strains. Okay, so we'll click OK, and we see Pathologic is now creating a new directory, and it's done. It's created a new PGDB. The PGDB is essentially empty. It contains a schema consisting of several hundred classes, but there are very few instances, and the database is basically empty at this point. It's been initialized. Now, after we run these various Pathologic commands, it will describe possible next steps here. And It'll also show you here our currently selected organism. Just as for the navigator, there's a notion of a currently selected organism for Pathologic. It tells us the status of the database, that it's in an initialized form. Also under the database menu, we could select a different database. We could reinitialize the database. If we thought, for example, our database was corrupted, we could reinitialize it. We can also here convert a file PGDB to MySQL. Okay, so those are the main operations. Typically, the next step that we do is to do what's called a trial parse, where what the software does is to read the one or more input files that we've provided and try to parse them, try to process them, and print a summary of what it's found. This particular bacterial genome is not closed. There are multiple contigs. The contigs are listed here, and for each contig, it's listing the number of genes it found, proteins, RNAs, and pseudogenes. And so this is an opportunity for a basic sanity check, that it's reading the input data appropriately. We used to have troubles where sometimes data would be missed, and so it's a good practice to look in your file, count up the number of genes and proteins and so forth available for one or two of these contigs. Just do some spot checking to make sure these numbers are coming out right, and that will give you some confidence that the file format is compatible with what the software is expecting. So doing a trial parse doesn't actually change the database. It just reads the input file without updating the database. But now, if we do an automated build, it will update the database. And when we do an automated build, we get this dialog that prompts us for various parameters related to the build. Progress windows are simply little windows that show us the status of the software, let us know if things are moving forward. And we're going to enable name matching and taxonomic pruning. Those are some aspects of pathway prediction. We're going to give a pathway prediction cutoff score of 0.15, which is the likelihood cutoff score. If a pathway has a likelihood value less than 0.15, it will not be included in the new generated database. You can change that value if you want to be looser or more stringent in your prediction. And this particular menu item indicates whether the software should bring into the new database pathways for this same organism that have been curated into the Metapsych database. So we'll check Metapsych to see if it has any curated information for the organism that we're processing. And so we can either not have it try to import such proteins and pathways, or let it do the import. Then down here, there are various refinement tasks that it will do. These are additional inference tools in addition to pathway prediction. There's the transport inference parser, which tries to infer transport reactions from the names of transporters. This box indicates whether it will try to create BLAST databases for running BLAST against the new genome. And if you open up each one of these triangles, it will explain in more detail. We're going to run transport inference parser, operon prediction, protein complex prediction, and we will generate a cellular overview. Operon prediction should only be run for microbes, not for eukaryotes. The protein complex predictor analyzes the names of proteins in the annotated genomes and attempts to infer protein complexes when those names indicate membership in a common complex. And the cellular overview diagram is the large metabolic map diagram that contains all of the pathways predicted for the organism. So at this point, I'm going to click OK, and it will begin generating the database for real now. It parses all the different replicons within that file, gives us essentially the same table we saw before, and it's now doing reactome inference. It's going through all the different enzymes in the genome and trying to match them up with the reactions they catalyze based on EC numbers and gene ontology terms, and as a last resort, using gene names. It's running its reaction inference now on 3,135 input proteins. At this point, it has finished its Reactome inference. It's now starting the Pathway inference, and it's just written a report file on Pathway inference. There are actually several report files written about Pathway inference that have slightly different information, and I would recommend opening up this report file. It also writes a name matching report file shown here that contains the results of the Reactome inference. I recommend reviewing that file as well. It's now deleting pathways that do not have sufficient evidence for their presence in this organism, and also cleaning up predicted reactions and metabolites that were part of those deleted pathways. It's now running the operon predictor. It's finished pathway inference and it started the operon predictor. It's now running the transport inference parser. It's now inferring protein complexes. Okay, it's saved the database. That means all the predictions it's just made have been saved in a persistent form in this file. It's now creating the overview graph, the cellular overview containing the metabolic pathway map. Now, why would we run Pathologic interactively as opposed to at the command line? It gives us more options. For example, those parameters that we set, we can change these various parameters and cutoffs. We can control which tools get run. The command line gives us less control over that. We can do the trial parse that the command line doesn't allow. So there are several reasons that we've provided the two different options. Okay, at this point, Pathologic has completed. So there are a number of things we can do at this point. One thing I'm going to show you is how to do some simple searches across the database. The database is now fully functional, although there are some more refinement aspects of Pathologic that I'm going to show you in just a few minutes. But for now, let's exit from the Pathologic tool. And we're now back to the navigator, and our new database is now listed here. I'm going to select that as my current database. We get various metadata about the organism here, various statistics on the different replicons. And if we want to enter a genome browser for that replicon, we simply click there, and we're in the genome browser now. We can zoom in on a particular gene to make it a bit bigger. We can move left and right. We can click on a gene page to see what is known about METF. Okay, this is the product name for METF. It catalyzes this reaction right here. If we click here, we'll go to a reaction page. You can click on a metabolite to go to the metabolite page, which lists all the reactions and pathways this metabolite is involved in. If I click here, we'll go to a pathway page, and if I do a right click, I can say show me this pathway in the cellular overview, and it did build the cellular overview as we saw earlier. So there's the pathway we just came from, and this is the desktop version of the cellular overview. We could run the software in web server mode as well if we wanted to interact with it through the web. We can mouse over a metabolite to see what that is and what pathway it's in. One thing to notice about the cellular overview diagram is that notice most of the reaction lines are fairly dark, but there are some that are very light. Well, those are, quote, pathway holes, like that one right there. That particular reaction, let's click here and show the pathway. Here we've shown that pathway in a pop-up window. And note that this particular reaction we were looking at, which was shown in a very light line, has no enzyme associated with it. This has an enzyme. There's the enzyme name and the gene name. This one has an enzyme name and a gene name, but the fact that there is no enzyme name and gene name shown means that no protein has been associated with this particular reaction in this organism. You know, we can also do searches using this menu. So I can search, for example, for L-arginine, and it will take us to the metabolite page for L-arginine. So as I said, the database is fully functional. But we can, at this point, make several types of improvements to the database. So let's invoke Pathologic again. And we can see that when we invoked it, it continued to use the same organism. So that's just a sanity check that this is still our current organism. Notice its status has changed from initialized to built because we have a built database. We could choose to reinitialize it to essentially delete everything that was just inferred and rerun the pathway prediction and so forth if we want, but we're not going to do that. What we're going to do is to run some of the refinements. Refinements include both inference tools like the transport inference parser, but they also include a number of interactive tools for reviewing and updating and correcting the information that was just inferred. So for example, if I click on assign probable enzymes, I get a tool for reviewing the enzyme reaction associations that were inferred by Pathologic and for correcting, changing, or giving my stamp of approval to these different inferences. So here it's listing in this tool enzymes that it was unsure about, either because it found their name to be ambiguous and thought that they could be associated with multiple possible reactions, or it simply wasn't sure which reaction to associate them with. Now, notice that some of these enzyme names are very nonspecific. They refer to general classes of enzyme activities, but not particular enzyme activities. We wouldn't expect these to be assigned to a particular reaction. But the software is more or less simply looking for all the proteins whose names end in ASE, with a few exceptions, that were not assigned to a reaction, and bringing them up here for us to review. And so, for example, if I click on this one, I get the probable enzyme status dialog, where I have the choice of either assigning this protein to a reaction or saying, don't show this protein in that table on the previous screen because either it's not a metabolic enzyme at all, or it has a very non-specific enzyme activity or some other reason. So there's our reason. So we're now not going to see that protein anymore, or it will be grayed out. Now I could also, for one of the ambiguous ones, I could go into this same dialog and choose one or more of these possible reactions. See, there are quite a few different reactions that the enzyme could have been assigned to. I can simply click one or two of these if I've done some research and determine which one it should be assigned to. Or again, I could say, don't show me this one in the future. As another example, I can go down here to this enzyme, click on it. Here's my probable enzyme status dialog. It's identified 3 possible reactions that this enzyme could be assigned to. And so let's say I've done some research and I've decided only the first one really makes sense for this enzyme based on sequence analysis that I've done or literature research about this enzyme. Often you'll want to do a PubMed search or a Google search on the enzyme name and see what you can find out about it. And often you can distinguish which reaction it should be assigned to. You can also, by the way, assign it to a different reaction, either an existing reaction in Metapsych, or you can create a new reaction to assign it to. But what we're going to do is simply assign it to the first reaction in the list. Click Assign. Now, in this new dialog, we can do things like look in the navigator at the reaction that we're going to assign it to. We can create a new reaction. We can edit the enzyme activity name, like maybe I don't want HEMJ to appear at the end there, so I'll remove that. We can enter new synonyms, enter a comment for why we made that assignment, and click OK, and OK again, and now we're back to this tool. And notice it keeps track here of how many proteins have been assigned to reactions, how many are ambiguous, etc., etc. And so we can exit from the tool, and we can enter a different refinement tool. Let's say I want to look at the transport inference parsers assignments. Well, what it's telling us here is that TIP was run in batch mode. So when we did the run of Pathologic, it did run this basic tool. And so I can either at this point abort and do nothing, or I can say, yeah, I want to review the results of that batch mode TIP session. And it's going to bring up a similar window to the one we just saw to let me review the different transport reactions that were inferred by the tip tool. In this case, there are 17, but it's only showing one of them. I think because for the 17, the tool is fairly sure that they're not of interest, that they had very non-specific transporter names, and so it's not really worth looking through them. Now, if I really want to see those, I could go under the filter and say specify the filtering criteria. Notice that we've told it by default to exclude the low probability transporters. If I unclick that, we now see a longer list. And so we can see most of these transporters have names simply like ABC transporter or amino acid ABC transporter that are very non-specific, and that's why they weren't shown. So let's turn that exclusion back on again, and let's click on the one transporter that's shown here, and we get a transport inference parser dialog, which lets us change the name of the transporter if we wish. It's showing us the subunits that were converted into a complex, probably because they're near each other on the genome, which, yes, we can confirm that by looking at these identifiers. Tell us these genes are very near each other on the genome. We can edit the transporter subunit structure if we wish. We can change the energy coupling mechanism to be a channel transporter if we wish, but it's pretty clear from the fact that this is an ABC transporter that it's ATP driven. This is the reaction that it's used from Metacyc to associate this transporter with, and it's going to customize that Metacyc reaction when it's imported into this PGDB by putting compartment information in front of each substrate in the reaction. So it's telling us that ATP in the cytosol plus methionine in the periplasm are transported to yield, well, ATP is dephosphorylated in the cytosol, the methionine is transported into the cytosol, and here's our phosphate in the cytosol. We could create a new transport reaction if we wish. Overall, this is fine the way it is, so we're just going to accept it. We could reject it if we want and make no assignment of a transport reaction to this transporter. So now the status is changed to accepted, and I'm going to save the database and exit from this tool. And it's telling me there's 6 unreviewed transporters. Are we sure we want to save and exit? And yes, we will. And there we go. So now one thing you might want to consider doing is it's sometimes worthwhile to redo the pathway prediction, because if we've assigned a number of additional enzymes to reactions, that means that there are now more reaction activities available in the database to support pathway inference. So some of the pathways that were predicted that were not predicted before because they didn't have enough evidence, they may now have more evidence for their presence in this organism. And so if we rerun pathway prediction, we may get slightly different results. Similarly, either because of the changed reaction assignments or because of now changed pathway assignments or transporter assignments, we may want to re-update the cellular overview and recompute that, rebuild that diagram, because that diagram may now be stale and reflect old information. So it is useful to rebuild that diagram periodically. So those are the basics to creating new pathway genome databases using Pathologic. There's a lot more information about Pathologic and this whole process in the Pathway Tools User's Guide. There's an entire chapter on running Pathologic.